So in this video, I'm going to talk about chemistry of nucleic acids. I'm Dr. Baskaran, Associate Professor, Department of Biochemistry, Sri Lakshminarayan Institute of Medical Sciences, Pondicherry. Specific learning objective. At the end of the session, this particular session, the learner should be able to describe nucleotides and nucleosides, purine and pyrimidine bases, functions of nucleotides, synthetic nucleotides and their function, structure and functions of DNA, structure and functions of RNA. Introduction, and this whole session will be based on this. Nucleotides or components of nucleic acids. So we talk more about the nucleotides. Free nucleotides have multiple functions like high energy compounds, metabolic regulators, etc. Synthetic nucleotides are synthesized or produced in the laboratory, and they are structurally modified, and they are used in oncology and clinical research. Nucleic acids are a repository of genetic information, and there are two types: ribonucleic acids, deoxyribonucleic acids. Let's start with the nucleoproteins. Nucleoproteins are conjugated proteins. That means they're complex protein or combination of protein and non-protein parts. In this case, nucleic acids are non-protein parts. The functions, they have a storage and transmission of genetic information. They are responsible for maintaining the genetic integrity of the species. Examples include eukaryotic chromosomes, ribosomes with RNA, viral particles. Chromosomes, we have a DNA and histones on the other basic proteins. They form chromosomes, the nucleus. Ribosomes with RNA, nothing but rRNAs. They have these ribonucleic acids with the various proteins, a polypeptide chain. Viruses, all viruses are nucleoproteins. Basically, they have one nucleic acids inside, maybe a DNA or RNA, and one envelopes, protein envelopes. So viruses are nucleoproteins. Nucleic acids are polymers of nucleotides, generally more than 50 nucleotides. These nucleotides are joined by phosphodiester bonds. That means the nucleotides are held together, connected by phosphodiester bonds. This one shows the phosphodiester bonds. In this case, here the phosphoric acids and hydroxides, the hydroxyl groups from the ribose, formed the ester bonds. There are two ester bonds, so therefore it is a phosphodiester bond. Now we'll focus on the nucleotides. Nucleotides are made up of nitrogen bases, either purine or pyrimidine, 5-carbon sugar, it's a pentose, either ribose or deoxyribose, phosphoric acids. We can say it's simply, or they are derivatives of nitrogen bases because they formed basically from nitrogen bases. And these are the two nitrogen bases. One is purine and the other one is pyrimidine. And this purine is a 9-member carbon compounds. Pyrimidine is a 6 carbon compounds. And it is basically heterocyclic compounds. And also the basic in nature is because heterocyclic means not only the carbons, they have besides carbon, they have other elements also like nitrogen, sulfur, etc. In this case, and the nitrogens are present in place of carbon, so they are heterocyclic compounds. On the purine basis, we have two basic these things, adenine and guanine. Adenines, you can say the 6 amino purine, while guanine is 2 amino 6-oxy purine. These two are major bases from purine ring system. While from pyrimidine bases, we have cytosine, uracils, and thymine. I'm not going to the individuals naming. For examples, 2-oxy, 4 amino cytosines. So we can say this way, Very simply, we can say cytosine, uracils, and thymine. Major bases, these 5 major bases, or you can say these 5 bases, adenine bases, are predominantly found in nucleic acids, and therefore they are called major bases. These are actually this adenine, guanine, cytosine, thymine, uracils. Among these 5 bases, thymine is closely present in DNA, while uracil is found in RNA. Sugar moiety or sugar residues, we have ribose or deoxyribose, the pentose sugar. In this ribose, it's a D-isomers, D-ribose, one should say, and 2-deoxyribose, 2-deoxy-D-ribose, and this deoxyribose, we have the second position, the oxygen is replaced by hydrogen, so that's why it is deoxyribose. Nucleosides, these purine bases or trivalent bases, when attached to sugar, simply you can say adenine bases when attached to the sugar, then they are called nucleosides. The sugar that can be either D-ribose or deoxy-D-ribose. They are joined by or connected by N-glycoside bonds, and therefore they can say they are also derivatives of glycoside compounds. They actually, from the purine is the ninth position, while in the pyrimidine it is the N1 position, where the it is basically beta and glycosidic bonds that only connects the bases to the sugar. This table shows the ribonucleotides. I'll just adenine. I'll just say this one example. Adenine, when with ribose, when attached to ribose, it becomes adenosine. Guanine, with ribose, forms guanosine. Uracils form uridine. Cytosine forms cytidine. While hypoxanthine and xanthine, these are the purine bases. Hypoxanthine, when it is ribose, it forms inosine. This is a compound formed during the synthesis of purine nucleotides. Xanthine with ribose forms on the zinc. This is actually formed during metabolism or breakdown of purine nucleotides. Deoxyribonucleosides, same. So adenine with the deoxyribose is formed deoxyadenosine. Similarly, guanine becomes deoxyguanosine. Cytosine becomes D-cytidine. Thymine, usually we say thymidine, but you want to be specific, you can say deoxythymidine. Nucleotides. These are the, like when nucleosides, when phosphorylated, they're called nucleotides. These nucleotides can exist in the mono, di, or tri phosphorylated forms. That depends upon the number of phosphate groups. 1 means mono, 2 di, and 3 is tri. So, for example, adenosine 5 monophosphates, or simply, or say to be particular, adenosine 5' monophosphate, AMP. The di and tri-phosphorylated forms for ADP and ATP. Here it shows the both nucleosides and nucleotides. The adenosine is nucleosides. This is a ribonucleotides. This when it's attached to phosphoric acids becomes adenosine 5' monophosphate or AMP. So that's a nucleotide. So we can add one more and 2 more phosphates. It becomes ADP and ATP. This one is nucleosides and nucleotides. Adenine becomes adenosine, and adenosine monophosphate AMP, that is nucleotides. Guanine becomes guanosine, and guanosine 5 monophosphate GMP. So things will go like this. This one is deoxynucleosides and deoxynucleotides. For example, adenine becomes deoxyadenosine, and deoxyadenosines 5 monophosphate TAMP. Similarly, other compounds will be formed. Nucleosides, nucleotides, actually this numbering system That's one is clockwise, other one is anti-clockwise in the energy basis. Besides, we have the prime rotation for ribose to indicate the position of the atoms in the ribose ring. So when it's, for example, when we say 5 prime, it is indicate the position on the ribose. For example, 5 prime AMP. Usually we drop the notation in the case of 5 prime because it's the most common, whereas in the 3 prime it is indicated always. So 3 prime AMP, it means the what is called the phosphate is on the third position. This is to avoid any confusion, between ribose and and nitrogen bases. They have the properties of absorbing UV radiation. So by these even nucleic acids, that's because of these nitrogen bases, they have conjugated double bonds. So, and because of this, they absorb UV radiation. The maximum absorption is at 260 nanometer, and that actually causes any kind of mutagenic effects. That's because of, again, UV radiation causes some chemical changes. That's what mutation in the DNA structure. That leads to mutation and cause genesis. Cyclic nucleotides here in this case. So far we have seen the phosphodiester bond between the two nucleotides, whereas the phosphodiester bond can be formed within the compounds, between 3 prime and 5 prime position, and these compounds are called cyclic nucleotides. We have two examples, cyclic EMP and cyclic GMP. They act as second messenger and involved in the signal transduction. That means they carry out the hormonal action. Cyclic AMP is a second messenger for many peptide hormones. For example, glucagon action is mediated by cyclic AMP. Cyclic GMP is a second messenger for nitric oxide and also atrial natriuretic hormone. This picture shows the cyclic AMP structure. The bond is the internal phosphodiester bond is between 3 prime and 5 prime position. Cyclic AMP. How it is formed? It is formed from ATP by the access of adenyl cyclase or adenylate cyclase, and is deactivated by phosphodiesterase. So this is once the any hormones, the case we take up that case of glucagon when it's induced through the receptors. We can talk something about phosphodiesterase. This one, which once the hormone access over, the cyclic AMP is destroyed by phosphodiesterase, and therefore the hormone action is over, terminated, whatever. But this phosphodiesterase is inhibited by certain compounds like caffeine and theophylline and all. These are present in the coffee and tea respectively. That's why when they inhibit phosphodiesterase, the cyclic AMP constant is enhanced, and therefore the hormonal actions is prolonged. Similarly, cyclic GMP is formed by the action of guanylyl cyclase and deactivated by phosphodiesterase. Functions of nucleotides. And these nucleotides are the building blocks of nucleic acids, both DNA and RNA. They also involve in the energy transaction. For example, ATP is high energy phosphate compounds. That only provides energy for all the function, mechanical actions, synthetic function, etc. Physiological mediator, for example, second messenger, cyclic AMP and cyclic GMP, components of cofactor, to be precise, coenzymes. For example, NAD, niacin, adenine dinucleotides. This one is a phosphates, NADP, FAD, flavin, adenine dinucleotides, FMN, that's a mononucleotides. This one is coenzyme A, that is actually pantothenic acids. In all these things, we have nucleotides, and also they act as allosteric regulator. For example, ATP, ADP, EMP, they actually regulates metabolism by acting as allosteric regulator. Now we'll see those are the general functions, but here we focus on the individual nucleotides. Here, adenosine derivatives, besides building blocks of nucleic acids, these involve energy transfer reactions. For example, ATP on hydrolysis releases 7.3 kilocalories, and that's used for all the like energy requirements. It acts as a second messenger, for example, cyclic AMP as a second messenger. This already mentioned, this acts as a second messenger for glucagon. This active sulfate, PAPS, phosphorodinosine phosphosulfate. This one is act as a donor for sulfate groups, active sulfates, for synthesis of mucopolysaccharides, glycosamine glycane, and also for conjugation reaction. Next one is active methionine, and here this is SAM. Yes, adenosyl methionine. This is actually donor for methyl group for the synthesis of methylated product. For example, the catecholamine epinephrine synthesized from norepinephrine after getting this methyl groups from SAM. And this methyl group also used for various conjugation reactions, that is from S-adenosyl methionine, SAM. In guanosine derivatives, this GTP used for protein synthesis, and also it's a second messenger, for example, cyclic GMP. It's a second messenger for nitric acids, and also HCl natriuretic hormone. Uracil derivatives. This is involved in the carbohydrate metabolism, particularly in the glycogen synthesis, ADP glucose, and also in the conversion of sugar from glucose to galactose. That is, for example, UDP galactose. In the conjugation reactions, for example, UDP glucuronic acids or uranic acids is used in the conjugations of bilirubin. That way, the bilirubin is detoxified in the excreted in the liver, making it soluble along with uranic acids. Cyclic derivatives, this is used in the lipid synthesis. For example, CTP diacylglycerol for the biosynthesis of phosphoglyceride, and also sphingomyelin. Minor bases. These bases are found in small quantities, and therefore they're called minor bases. They are involved in the recognition and protection of these molecules. For example, 5-methylcytosine is found in prokaryotic and eukaryotic DNAs. 7-methylguanine and is found in eukaryotic mRNA. And 7-methylguanine is a cap, is found in the mRNA, 5' position. 5-hydroxymethylcytosine is found in bacteriophage. And also pseudouridine is not here, is found in the tRNA. Free nitrogen bases, they are intermediate in the catabolism of adenine and guanines, for example, liposamine and xanthine, and finally it's catabolized to uric acids. Methylated nitrogen bases, they are plant products, and also they are methylated xanthine. For example, caffeine. Caffeine is a trimethylated xanthine. It's found in coffee, theophylline in tea, and theobromine in cocoa. These two are dimethylated xanthine. Synthetic nucleotides are basically analogs of nucleotides, nucleosides, and bases, and even sugar, the ribose. Here they are sexually modified, and they have wide applications, medical and research. They are used as anti-tumor, anti-cancer, and antiviral agents. For example, and as anti-tumor agents, 5-fluorouracil, 5-iodouracil, 6-mercaptopurine, and 6-thioguanine. As antiviral agents against HIV, azidothymidine, and deoxyribonucleotides, used in the treatment of gout, allopurinol, and as immunosuppressant, azathioprine is used in the transplant to prevent any tissue rejection. Polynucleotides are formed by the condensation or combination of 2 or more nucleotides linked through a phosphodiester bonds. The primer structure or the basic sequence is as follows. This is 5' to 3'. This shows is 5', there's a phosphate, at the 3' it's free. This nucleotide, polynucleotide runs in the directions of 5' to 3' directions. It's always written this way. So next one will show the actual things like these well sets can be represented ACGTE, that is a 5' to 3'. That's a phosphodiester bond between these. This one is a phosphodiester bond. And this is actually shows from the DNA and RNA, the phosphodiester bonds. Now we'll take up the nucleic acids. Nucleic acids are polymers of nucleotides. There are 2 types: ribonucleic acids, polymer of ribonucleotides; deoxyribonucleic acids, DNA, polymer of deoxyribonucleotides. Function: storage and expression of genetic information. History, when we talk about this nucleic acids, in the 1868, measure isolated nucleic acids. It was named as Nucleic. In 1882, Altman introduced the terms nucleic acids. In the early 20th century, Koshal describes the 4 bases of nucleic acids. Later, Thomas Morgans found chromosomes contain genes in a sequence order in Drosophila. Northrop found virus activity was due to nucleic acids. Avery and his colleagues like McLeod and McCarthy, they found non-virulence to virulence in bacteria. Charcot put forward the base pairing rule in 1950, the middle of the century. Later, Maurice Wilkins and his scholar Rosalind Franklin, they formed the X-ray crystallography. And with that X-ray crystallography, these 2 people, Watson and Crick, one is physicist, the other one is biologist. They deduce the structure of DNA, what is called now called Watson-Crick model of DNA or bDNA. All 3 actually, Rosalind didn't receive Nobel Prize because he died before receiving that. So these 3 people shared the Nobel Prize later, almost 10 years later. It was the structure was found in 1953, and they received Nobel Prize in 1962. That let's take a look at the structure of DNA. Watson-Crick DNA or bDNA. This, according to this Watson-Crick model, The DNA is a right-handed double-stranded helix. It's helical molecules containing 2 strands, and the strands are antiparallels. That means they're running in opposite direction. The sugar and phosphate backbones of each strands coil around the outside of the helix, while the nitrogen bases point towards the center of the helix. That means they're located inside the molecules. That's maybe because of the hydrophobic in nature, while the sugar and phosphates are outside because of the hydrophilic in nature. The diameter is 2 nanometers. One complete turn is called a pitch that contains 10 base pairs. The length is 3.4 nanometer. That means each base pair is kept at the distance of 0.34 nanometers. The strands are held by hydrogen bonds between complement bases. A always pairs with the T with the 2 hydrogen bonds. G always pairs with the C with the 3 hydrogen bonds. Being a double stranded, it obeys Chargaff's rules. It states that in a given DNA molecules, the amount of adenine equals the amount of thymine, and the amount of guanine equals the amount of cytosine. There are 2 distinct groups. One is major, one is minor. Major is 1.2 nanometer, whereas minor is 0.6 nanometer in depth. So they are involved in the interactions with the enzymes during replication and transcription, etc. One of the standards template where the information is found, the other one is called coding strands. This actually picture shows the Watson-Crick DNA model or BDNA. This is one complete distance turn, which is just given in the Armstrong units. So let's go to this other one. Here is a 3 points. 1, 10 should be 10 base pairs. Here we can count this 10 base pairs 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So that is 10 base pairs in complete turn. So that means it's a Watson-Crick model. What are the types of DNA? There are 6 types of DNA: A, B, C, D, E, and Z. Among them are 3 are most common. And the BDNA or the Watson-Crick model is found in the physiological condition or the biological condition. This is normally what we have. ADNA is right-handed but dehydrated form, contains 11 base pair per turn. ZDNA as the left-handed helix with 12 base pairs turns. This is not actually usually present, but formed during the sometimes only when the some gene expression and all. This shows the characteristics of these 3 DNA molecules. So we can check with this. The BDNA is diameter is 2 nanometers, base pair is 10 per turn, and the degree of rotations 34.6, while distance on complete turn is 3.4 nanometer, and between the base pairs, the base is kept at the distance of 0.34 nanometers. So they will be changed the other things because this is the dehydrated one, this is the left-handed, formed in particular times. Denaturation when a solution of DNA is heated, that heat energy pulls the strands away, pull apart the strands. That is called denaturation. The temperature at which the strands are separated is known as melting temperature. Usually, the DNA rich in GC pairs have higher melting temperature because there are 3 hydrogen bonds. So they need more energy for separating the 3 hydrogen bonds. Denaturation, when the when the DNA is denatured because the heat and it's cooled down slowly because the complementarity, the strands will come together and form again the original DNA molecules. And this process is known as annealing or denaturation. This picture shows the denaturation followed by denaturation of DNA. Ribonucleic acids, RNAs, as a polymer of ribonucleotides linked through again 3' to 5' phosphatase bonds. They should not be confused with the direction of the molecule. Direction is always from 5' to 3'. The bond is formed from 3' to 5' here. That is. Some differences are there between DNA and RNA. RNA differs from DNA as like RNA is single stranded as a very common things. The sugar is, in RNA is ribose, or not deoxyribose. In the DNA, it's a deoxyribose. RNA contains uracil, while DNA contains thymine. The most important difference is RNA can be hydrolyzed by alkali, whereas DNA resists hydrolysis because the deoxyribose. That means DNA is more stable than RNA. That may be because evolutionary, because DNA is nature has chosen the DNA as genetic material for higher organisms. So that is because of stability. DNA is more stable than the RNA. Types of RNA. In all prokaryotic and eukaryotes, we have 3 major classes of RNA exist. Say there are 3 major RNA found. RNAs are found. These are messenger RNAs, mRNAs, transfer RNAs, tRNAs, ribosomal RNAs, rRNAs. The other RNAs or ribonucleic acids found are snRNA, miRNA, micro, small interfering RNA, siRNA, heterogeneous nuclear RNA. While heterogeneous nuclear RNA is give rise to other, like RNAs that's processed into messenger RNA. While these small, micro, and small interfering RNAs now involves non-coding RNAs involved in the regulatory process of transcription and translation. This one is a messenger RNA that's in the cell form by the transcription undertaken to this within the nucleus, which means it's about 5% of RNAs in a cell. It's most heterogeneous because one polypeptides corresponds to one messenger RNA. So it's most heterogeneous in nature. Actually, it carries information for protein synthesis. That means transport message from gene to the protein synthesis machinery. The 5' terminal or the 5' end is capped with 7 methyl guanosine triphosphates. While there's this one is actually involved in the recognition of mRNA by the translating machinery, and also it stabilizes mRNA by protecting it from 5' exonucleases. While 3' end is containing some around 200 adenylate residues, what is called polyatyles. This also involved in the production of these RNAs, messenger RNAs from exonucleases. Histone and interferons do not have polyatyles. That's one major things. Well, this one is messenger RNA, so in this cap. 7 methyl guanosine triphosphates, while it's a polyatyl. Messenger RNA is actually formed by the process of transcription or formed in the transcription. This sequence carried on mRNA is read in the form of codons. That means codons present in the messenger RNA. And it's a triplet 3 nucleotides. mRNA is usually formed after processing of endogenous nuclear RNAs. Functions: transfer of information from genes to the translation. So this is actually just we mentioned about endogenous nuclear RNAs, hnRNA. Actually, this is immediately, it's immediate product of gene transcription. The transcription process, endogenous nucleus RNA is formed. This is actually undergoes various, like process, modification, process, and process 2. Finally, it's process to mature mRNA. Various processes, splicing, etc. Contains various like introns. So these are removed, spliced to form the matured messenger RNA. That's actually contains the codon for protein synthesis. Now we come to transfer RNA or tRNAs. Transfer RNAs are the smallest of 3 major species of RNA molecules. They have 75 to 95 nucleotides residues. They transfer the amino acids from cytoplasm to the protein synthesis machinery, and therefore it is called as tRNA because transporting amino acids. They are also soluble and also called soluble RNAs or sRNA. They are also called adapter molecules because they act as adapter for protein synthesis. There are at least 20 species of tRNA, one corresponding to each of the 20 amino acids for protein synthesis. The primary structure of RNA on this tRNAs sequence of nucleotides, but it's fold itself to form a secondary structure. The secondary structure is, like it's a clover leaf-like structure, and the structure is stabilized by hydrogen bonding between the bases. So the secondary structure of tRNA is a clover leaf structure. All tRNA contains 4 main arms with 1 extra arms. Based on the extra arms, we have 2 classes of tRNA, class 1 and class 2. And the 4 arms are acceptor arms, anticoda arms, DHU, and TCIC arms. Now let's start with the acceptor arms. It's at 3' end. It has 7 base pairs. And it ends with unpaired 3 sequences, 3 bases, cytosine, adenine at the 3' end, CCA, that is. This only forms, with bond with amino acids for transportation. Once it is attached to amino acids called amino acyl tRNA. Anticoda arms is opposite to the acceptor arms. It has got 5 base pairs now. It recognizes the triplet codon present in the mRNA. That means there's a complementarity. Because of this complementarity, it forms, it's easily bind with the codon and forms hydrogen bonds during the protein synthesis, while the other acceptor arms contain the specific amino acids for protein synthesis. While DHU arms, it has a diuridine. That's what it shows this. It has 3 to 4 base pairs. It's involved in the regulations. Maybe it's involved in the regulations for the enzymes, amino acyl tRNA synthetase, that adds the amino acids to the acceptor arms. D-cyc arms. This arm is opposite to DH arms as it contains pseudouridine. It's involved in the binding of tRNA to the ribosomes during protein synthesis. Structure of, in continuation of structure of RNA, the extra arms or variable arms, more than 70% of tRNA molecules have short extra arms, about 3 to 5 base pairs, and that is actually class 1, while the rest of them are, or maybe that's class 2, have the extra arms have 13 to 21 base pairs in length. This picture shows the structure of secondary structure of tRNA. And this molecular transfer from E5' to 3' is free at the 3' it's E, accept arms, some base pairs. This opposite is anicodon. This is actually, we have 3 loops on extra arms. This is corresponds to the codon of the mRNA. While this is a G arms, DHU, while this is D-cyc arms. Functions. It's an information adapter molecule. That means that is transport of amino acids. It's involved in the transport of amino acids for protein synthesis. Now we come to ribosomal RNAs or RNAs. They are majority of among the RNAs in eukaryotics. More than 80% of all eukaryotic RNAs constitutes ribosomal RNAs. It contains 2 nucleoproteins subunits, the larger subunits with 60S and smaller subunits with the 40S. The 60S subunit contains 5S RNA, 5.8S RNAs, and 28S RNAs, and with 50 specific polypeptide. The S stands for Swarbath units. It's a unit of sedimentation rate. The 40S subunit is smaller and contains a single 18S RNA and approximately 30 polypeptide chain. All of the ribosome RNA molecule except the 5S RNAs are processed from a single 40S precursors RNA molecule in the nucleus, while 5S RNA is independently transcribed. This shows the ribosomal RNA. This is a larger unit. This is actually formed to smaller units, 60S and 40S. During, translations, they form the larger units. These 60S is 28S RNA, 5.8, 5S RNAs with around 50 proteins, polypeptide chain, while 40S is 18S RNAs with 33, around 40 proteins. Functions, they are necessary for ribosomal assembly and also play key roles in the binding of mRNA to ribosomes and translation. Some of the RNAs act as enzymes involved in the peptidyl transferase activity. That's called ribozyme. Here we stop. Thank you.